Hi everyone, my name is Mr. Barlow and welcome to episode 32 of the VCE Biology podcast. This episode covers part of Unit 4, Area of Study 1, and I'll be talking about genes, chromosomes, alleles, genotypes, genetic inheritance, and pedigrees. So, parents have children, and more often than not you'll find children look a lot like their parents. And that's because parents pass their genetic material, their deoxyribonucleic acid, or DNA, down to their children. And that's why children look like their parents. Parents pass their DNA down to their children in very special cells called gametes. So in humans, this is basically a sperm cell from a male and an egg cell from a female. So gametes are also known as sex cells, and they're sometimes referred to as germline cells too. So gametes are very special. They pass DNA down from parent to child, and gametes are different from all the other cells in a multicellular organism. All the other cells are called somatic cells, so they're very different from sex cells. So DNA contains genes, and genes carry instructions basically to make an organism what they are. So these instructions can be called the organism's genotype. So a genotype is the characteristics of an organism's DNA, whereas a phenotype is the way that genotype is expressed, either physically or physiologically. So for example, I might have some DNA which controls the development of my eyes, but my phenotype might be, I have blue eyes. Now in cells, DNA is organised into these really big structures called chromosomes, and each chromosome's got a number of genes on it. Normal humans have got 46 chromosomes in total, and in fact they've got 23 pairs. They get 23 from their mum and 23 from their dad. So 46 chromosomes is also referred to as the diploid number, which is often written as 2n. So n is the 23 from the mum and 23 from the dad. 2 times n is 46. Gametes, or sex cells, contain half the diploid number. So they just have n numbers of chromosomes. So in humans, that's 23 chromosomes. So somatic cells, which contain all 46 of the human chromosomes, are diploid, and sex cells, or gametes, which contain half the number of total chromosomes, are called haploid. Now, when scientists want to analyse a person's chromosomes, they basically organise them into what's called a karyotype. And what they do there is they put all the pairs of chromosomes together. So they put the two chromosomes together that decide a person's sex, the X and Y chromosomes. So if you're a female, you get 2 X chromosomes. If you're a male, you get an X and a Y sex chromosome. And then they also get the other 22 pairs or autosomes together. So they get the sex chromosomes together and the autosomes together into what's called a karyotype. Now, different autosomes can be distinguished by the way they look. So they've got different sizes, some are big, some are small. The position of the centromere is different in different chromosomes, and they've also got different patches of light and dark bands on them. Now, if you've got two chromosomes which are a matching pair, they're said to be homologous. But if you've got two chromosomes which aren't matching, they're said to be non-homologous chromosomes. So we put chromosomes in their pairs, in their homologous pairs. Now, when gametes are formed via meiosis, occasionally changes or abnormalities can occur involving chromosomes. So, for example, numerical abnormalities, also called Aneuploidy, is basically an abnormal number of chromosomes. And so there's a couple of different types. It's basically when someone's got not enough or too many chromosomes. So, for example, if you're supposed to have 2 copies of every chromosome, if you've got 1 less chromosome, you'll have only 1 chromosome for 1 pair. And this is called monosomy. So you're missing 1 chromosome, basically. You should have a pair, but you've only got 1. Another type of aneuploidy is trisomy. So tri is like tricycle, means 3. And trisomy means that instead of having 1 pair of chromosomes, you've got 3 chromosomes. There's also conditions called tetrasomy, which is instead of having 2 chromosomes, you've got 4 chromosomes. And there's some examples of actual diseases which are caused by aneuploidy or numerical abnormalities in chromosomes. The most famous disorder is Down syndrome, which you may have heard of. Basically, people with Down syndrome have got 3 copies of chromosome number 21. So they've got a total of 47 chromosomes in total. They've got their regular 22 pairs of autosomes, their 2 sex chromosomes, but they've also got an extra chromosome number 21. This actually happens because in either the mother or the father, when gametes are being formed, chromosome 21, it's called non-disjunction, so basically chromosome 21 doesn't split into 2 different cells. Both chromosomes go into the same cell, and if that's a sperm cell, say, if that sperm cell happens to fertilise an egg with the normal number of chromosomes in it, those 2 copies of 21 from the sperm cell will join together with the 1 normal copy of chromosome 21 from the mother's egg to form an organism with a total of 3 copies of chromosome 21. So that's Down syndrome, that's trisomy. An example of monosomy, so where an individual's only got 1 chromosome when they should have 2, is Turner syndrome, and that's basically where a female is born with only 1 sex chromosome. So there's some numerical abnormalities in chromosomes. Another type of abnormalities is structural abnormalities to, 1 chromosome. So this is, when 1 chromosome, the structure is altered. And there's lots of different types of structural abnormalities. For example, there's deletions, and this is where 1 portion of a chromosome is just missing or it's been deleted. There's duplications. So this is when a chromosome is being replicated. A portion of the chromosome is duplicated, so you've got a person will have extra genetic material. There are things called translocations, and this is when a portion of 1 chromosome is transferred to another chromosome. So 1 chromosome will end up a bit shorter and another chromosome will end up a bit longer. Another abnormality is inversions, and this is where a bit of chromosome has broken off, turned upside down and then reattached. So the material is inverted. And at the breakpoints, 1 or 2 genes might have been affected. There's an abnormality called a ring, and this is where a portion of chromosome has broken off and formed a circular chromosome or a ring. And this can actually happen without losing genetic material, but when an individual with a ring goes to reproduce, it can cause some problems. So in fact, all of these structural abnormalities can cause problems to the individual who has those structurally abnormal chromosomes, but also if those people reproduce and pass on those abnormalities, they can be passed on down the genetic line. Interestingly enough, actually, though, structural abnormalities is one form of evolution. So the way chromosomes change over time is actually, it's part of the way that new species form. So although generally we see it as a bad thing to have, a structurally abnormal chromosome, it's actually how species evolve to form brand new species. Now I've already talked about how in humans, and in fact in other mammals, if you've got 2 X chromosomes, it means you're a female, and if you've got an X and a Y chromosome, it means you're a male. But why does that happen? Why is XX female and XY male? Well, it turns out that on the Y chromosome, there's a very special gene called the SRY gene, which stands for sex determining region on the Y chromosome. Basically, if you've got the SRY gene in a mammal, you become a male. If you don't have the SRY gene, you become kind of like the default, which is a female. But not all organisms have the same way of determining sex. For example, in birds and some reptiles and amphibians, they have what's called a WZ system. So what happens is male birds have 2 Z chromosomes and female birds have 1 W and 1 Z. So it's a bit flip-flopped from the mammalian system. Another different way of sex determination is called, as opposed to genetic sex determination, is environmental sex determination. And this happens in some reptiles. And basically, the sex of the offspring depends on the incubation temperature of the eggs. So, if the eggs are incubated and it's really hot, the organism becomes a male. If the eggs are incubated and it's really cold, the organism becomes a female. So, an organism's sex can be determined in lots of different ways, not just the way we normally think about it, XX, female, XY, male. So you might have noticed whenever you look at two brothers who've got the same parents or two sisters who've got the same parents, they never look exactly the same. Except, of course, if they're identical twins. But generally speaking, they never look exactly the same. And that's a bit weird, isn't it? Because they got the same DNA from their mum and their dad, didn't they? Well, of course, it turns out they didn't. So they don't have exactly the same phenotype. They don't look exactly the same because they don't have exactly the same genetic material. They don't have exactly the same genotype. And what happens is, in meiosis, when gametes are formed, the chromosomes from both the dad and the mum are mixed up a little bit. So two siblings are never going to get exactly the same mix of chromosomes from either their mum or from their dad. And so this mixing up of the chromosomes is called recombination, and it happens in two ways. The first one is called independent disjunction, and basically what this means is, if the dad has got two chromosome number ones, which he does, he's got a pair, a homologous pair of chromosome number one, well, one offspring of that dad might get the first of that chromosome pair, but another offspring might get the second of that chromosome pair. And of course, we've got 23 pairs of chromosomes, so you never know which one of the two you're going to get, and that's why offspring are different. The other thing that happens is called crossing over. And in meiosis, so when sex cells are being generated, when gametes are generated, the two homologous chromosomes line up together, and what happens is sometimes some material from one chromosome switches with the same material from the other chromosome, and the two chromosomes aren't exactly the same. So crossing over actually results in chromosomes from parents going to offspring that are totally different from the chromosomes that the parents themselves actually had. So some of the genetic material from homologous chromosomes switches over. So you've got these two things that happen, crossing over and independent disjunction, and these happen in meiosis, and as a result it produces an enormous range of variability in the offspring produced by sexual reproduction. So if you've got the male and the female and both of their chromosomes are changing around via crossing over and independent disjunction in meiosis, you can see that once the sperm and the egg meet, there's again a great many possibilities for a totally different offspring to be found. And that's one of the big benefits of sexual reproduction. As we know, asexual reproduction results in offspring that are genetically identical, but sexual reproduction results in offspring which have a wide range of genetic variability. And this is really important for the survival of the species. So, chromosomes are made of DNA, and DNA has on it genes. So genes are the instructions to make living things. What they do is they control basically the structures and the functions of organisms. So for example, the human genetic material, human DNA, has about 20,000 to 25,000 different genes. Genes basically encode for proteins. They make a living thing what it is. They have all the instructions. Now, different genes have different positions on different chromosomes. And the position of a gene on a chromosome is called its locus. So a gene's locus tells us the precise position that gene is found on a particular chromosome. Now, genes on one chromosome, so if you've got two different genes on the same chromosome, they're actually members of what's called the same linkage group. So in meiosis, generally speaking, if you've got two genes which are right next to each other, more often than not, those two genes will end up in the same gamete. So that's when two genes are said to be linked. Also, interestingly, a very small number of genes exists in the organelle called the mitochondria. And these genes are called mitochondrial genes, and so they're different from chromosomal genes. They're actually highly conserved, so they're very similar across even different species, mitochondrial genes. So genes are located on DNA, and DNA forms chromosomes, and we've got two copies of each chromosome, of each homologous chromosome in our cells. So in fact, we've got two copies of each gene in each of our cells. But in fact, one gene can actually have slightly different versions or different forms. So the different versions of the same gene are called different alleles. So for example, the gene for seed shape in pea plants exists in two forms. One form of the gene, or one allele for the gene, encodes for round seed shapes, and we can call that allele capital R. And the other allele codes for wrinkled seed shapes, and we'll call that one lowercase r. So you can have different alleles of the same gene. In this case, capital R and lowercase r, and other genes maybe we'd call the different alleles big B and little b, or capital T and lowercase t. So genes have got different alleles or different versions. Now humans are diploid, so they've got 2 copies of each autosomal chromosome, and they've also got 2 copies therefore of each autosomal gene. Now if a person's got 2 identical alleles of the same gene, they're said to be homozygous. If a person's got 2 different alleles, they're said to be heterozygous. But of course also lots of genes are located on the X chromosome, and these genes are said to be X-linked. There's also genes on the Y chromosome. Those genes are said to be Y-linked. Now, because males have got the Y chromosome, but they've only got 1 copy of the Y chromosome, males are said to be hemizygous, so that means they've only got a single copy of the gene instead of 2. And males are in fact hemizygous for both the X and Y-linked genes, whereas females are obviously heterozygous for genes located on the X chromosome. Interestingly though, something called X inactivation happens in cells of females, and what happens there is that only 1 chromosome is active in any somatic or body cell. So females don't get like a double dose of the genes on the X chromosome because only 1 of their X chromosomes is active in each cell, and it's a random, either one of the 2. So that's X inactivation. So we have 2 copies of each chromosome, and therefore we have 2 copies of each gene. And genes can have different alleles. So in theory, it's possible for us to have 2 different alleles for the same gene. And so genes are basically instructions which tell a cell what to do. So what happens if you've got 2 different alleles or 2 different sets of instructions telling a cell what to do? Well, there's a couple of things basically. One of the things which can happen if you've got different alleles is something called complete dominance. And this is where one allele is totally dominant, so that one gene is totally dominant over the other allele or the other gene. So there's a dominant gene and there's also a recessive gene when you're heterozygous. So for example, if we were capital A for one particular gene and we're also lowercase a, so we had the dominant capital A gene and the recessive lowercase a gene, the phenotype, or the thing which would be expressed, would be the characteristics of the capital A gene. So it's possible for one gene to be dominant over another gene in a heterozygote, and in those cases, the recessive allele, if a person's heterozygous with a dominant gene and a recessive allele, they're said to be a carrier for the recessive allele. Now, another way that different alleles can have a relationship with each other is something called codominance. So codominance occurs when both of the alleles are visible in the phenotype, so both of the alleles are expressed. A good example of this is blood grouping, so you can have the A allele and the B allele, and then your blood group is AB. So if you've got different alleles, one thing that can happen is one allele can be dominant over the recessive allele. Another thing that can happen is codominance, in which case both of the alleles are expressed in the phenotype. Now, it's actually really important to say that it's not just an organism's genotype or its DNA which makes it what it is, which makes its phenotype up. Many environmental factors also contribute to an organism's phenotype. So it's not just the genes, it's also the environment. And this is called it's nature versus nurture. So nature is the DNA, the genes, whereas nurture is the type of environment that an organism was brought up in. So it's not just DNA, it's not just nature, it's also environment, it's nurture. And both of those things together make an organism what it is. Now, getting back to alleles, in meiosis, one allele on 2 homologous chromosomes will separate into 2 different gametes in meiosis. So let's say we had 2 parents, and the dad was heterozygous for one gene, and he had big A and a little a, were his 2 alleles for one gene, and they both went into 2 different sperm cells. And let's say the mum was also heterozygous for the A gene, and she also had one big A and one little a gene, and so her egg cells ended up with some of them with a big A and some of them with a little a. What could happen when those 2 parents reproduced is that the offspring could get a big A from the dad and a big A from the mum. So that would make it homozygous for the big A gene. But it could get a little a from the mum and a little a from the dad, and that would make it homozygous for the little a gene. Or it could get a big A from the dad and a little a from the mum, or a big A from the mum and a little a from the dad, making it heterozygous for this A gene. And what we can do is we can draw things called Punnet squares, which tell us the chances of offspring having particular characteristics depending on the characteristics of their parents. And so a Punnet square actually shows the chance of each possible outcome in a mating or in a cross, not what actually will happen. So if you only have one offspring, only one of those chances is going to actually eventuate in a particular mating. Now, if we create a Punnet square and look at a cross involving a single gene, so just the A gene, this is called a monohybrid cross. But we can also create Punnet squares for what's called dihybrid crosses. And this is where we look at the way two different genes are segregated during meiosis to form gametes. And then we do, a Punnet square, and we can look at the chances of each different type of offspring being produced according to, the percentages of those two genes. So monohybrid cross is where you just have a look at the way one gene will work, and a dihybrid cross looks at the way two genes are going to, interact together. Now, one other type of cross is called a test cross. So this can be either monohybrid or dihybrid, but it basically involves one of the parents that's homozygous recessive. So it only carries genes which aren't going to be expressed if the thing it's crossed to has a dominant allele. Now, genes on different chromosomes during meiosis, they are sorted independently of each other. So where one goes, which gamete one gene goes into, has no bearing on where another gene goes into. But if two genes are on the same chromosome, they're said to be linked. So two genes on the same chromosome don't behave independently of each other during meiosis, and two genes which are on the same chromosome tend to be inherited together. So they tend to go into the same gamete and then form the same organism. But sometimes two genes on the same chromosome will go into different alleles if they've crossed over during meiosis. Now, if two genes are on the same chromosome, but they're very close together on the same chromosome, the chance of the crossing over point being between the genes is actually quite small. Whereas if two genes are on the same chromosome, but they're very far apart from each other, the chance of those two genes being separated by crossing over in meiosis is actually quite large. Now, what we can do is we can perform test crosses on organisms to find out, A, if genes are linked to each other, if they're on the same chromosome, but it's also possible to estimate the distance between genes on the same chromosome. And we actually estimate that distance in a unit called a map unit. So we can figure out, by crossing individuals, we can figure out how far apart genes are on the same chromosome, if they're close together or if they're far apart. So this podcast's been all about genetic material and how it works and how it's passed on and things like that. Well, another way to illustrate how genetic material is passed on through the generations is to draw what's called a pedigree chart. And this shows the phenotype for an organism and all its ancestors. Now, the pattern of inheritance in a pedigree chart can actually give us information about the characteristic that's been inherited. So, for example, a characteristic can be autosomal dominant, and in a pedigree chart, this is shown by both males and females having the characteristic, and all individuals will have at least one affected parent. So because it's dominant, the parent has to have it to give it to the offspring. Pedigree charts can also show autosomal recessive patterns. So again, both males and females can be affected, but two unaffected parents can have an affected child. Also, all the children of two people with the condition must also show the condition. So if both the mum and the dad both have an autosomal recessive condition, then all of their children will get those recessive genes and the children will have it too. Also, in autosomal recessive inheritance, the condition may not be present at every generation level in a pedigree. Another type of inheritance, pedigree pattern is an X-linked dominant pattern. And what happens here is a male with the trait passes on to all his daughters and none of his sons. So males pass Ys onto their sons, so none of their sons get it. And if a female has an X-linked dominant characteristic or condition, then if she's dominant for both of her X chromosomes, she'll pass it on to all her kids, but she may be heterozygous, so she may pass it on to some of her daughters or some of her sons, but all of her children will get an X chromosome from her. Now, another type of pedigree pattern is the X-linked recessive pattern, and in the recessive pattern, all the sons of a female with the characteristic, so female's got both recessive genes if she's got it, all of the sons will have the condition because they've only got one X and they get it from their mum, and all the daughters of a male with the characteristic will be carriers of the trait or the characteristic, but they'll not show it. They won't have the phenotype for it. So the male will give them, the carrier will give them one allele, which makes them a carrier, but unless they get the other recessive allele from their mum, they won't show that characteristic. And that brings episode 32 of the VCE Biology podcast to a close. I'm Mr. Barlow, and thanks for listening.